In the previous videos, we have discussed that how this TGF beta plays a role in the case of metastasis of the tumor cells. In this video, we will discuss how this TGF beta is also related to the glycans that are expressed on the TGF beta receptor uh, that are found on the cell surface membrane that we can see here, and how these glycans play important role in the metastasis. We will also discuss that thing in brief, not in details. Yeah, so these are the TGF beta receptors that are shown here. It is TGF beta 2 receptor, TGF beta 1 receptors, and there are other receptors also. And one of the important thing that this glycan structure does, glycan or carbohydrate sugar structure does, is it helps this TGF beta receptor to expressed on the cell surface membrane, or in other terms, these TGF beta these glycans that are expressed on the TGF beta receptors helps them to be secreted on the cell surface membrane. As we know that there are some signaling proteins that needs to be present end of this at the end of this post translation modification that is in the Golgi body, from where this trans Golgi network will divert the cells based on the signal proteins that are present, and these glycans will act as a signal proteins, as act as a signals for this TGF beta 2 and TGF beta 1 and other co-receptors, and helps them to express on the cell surface membrane, not on the other organelles within the cells. So this is one of the important roles of these glycans that are present on the TGF beta receptor. Other things the glycans plays is that it forms this lipid raft, and this lipid raft consists of these cholesterols that are present on the cell membrane, uh, and also these glycan structures, or more specifically this glycose phospholipid that are um, present on the cell surface membrane and close to these TGF beta receptors, and this along with cholesterol and these TGF beta receptors formed a lipid raft, and this helps in the TGF beta receptor and TGF beta interactions. And there are other kinds of configuration of these TGF beta receptors are also found that can be seen here. In this case, the what this glycan does is glycan helps this TGF beta interaction with this TGF beta interaction with this TGF beta receptors by preventing this TGF beta receptor and this beta glycan interactions. This as these glycans are present in between these receptors and beta glycan, the beta glycan will not be able to interact with this TGF beta receptor, and thus these glycans, however, are arranged in a way that the it will not interfere this TGF beta to interact with this TGF beta receptors in the normal case. And so this is just a basic idea how this receptors helps in this TGF beta and TGF beta receptor interactions. And the second figure or panel, or the figure on the bottom, will provide us the information that whenever this TGF beta induced or uh, interact with the cells, or more specifically tumor cells, it will induce the mesochemical like properties, or the epithelial cells will interact with TGF beta, it will induce mesochemical properties. And what are the changes that occurs in the case when the cells are in the when the cells acquire this mesochemical characteristic that is shown here. So when the cells are in the epithelial, uh, when the cells shows the epithelial characteristics, these are the glycans that are usually found, that we can see here, that most of them are these N glycan structures, such as these are the complex structures, and these are the high mannose structures, oligomannose carbohydrate structures. However, when these mesochemicals like properties are induced by EMT, there is a change in the glycosylation that can be seen here. And some of the major changes are shown by this core, and the changes are shown by this red square, and some of these changes are such as this addition of this core glycosylation and this S Lewis and S Lewis X and Psi Lewis A structures, and the silation and this Lewis Y structures are also starts to occur. That helps in the mesomechicals, that helps the cells to, or that are expressed when the cells acquire the mesomechanical properties, and this branching of these glycan structures are also increased. That can be seen here in this example, I branching, and with this alpha 2,6 link of the sialic acid. So these are some of the changes in the glycan structures on the cell surface membrane that are expressed whenever the cell acquires their mesomechanical properties. So one of the application of this could be that if the cells has this kind of glycan structures, then they might have these mesomechicalized properties, which can be further verified using other reporters such as E-Catherine levels of the cells, N-Catherine levels of the cells, which is the marker for the mesomechicals properties, and E-Catherine is the markers for epithelial like cells. And there are other markers are also that are mentioned here. So these markers can be visualized in order to further validate whether the cells has their mesomechicals like properties or not. So these are how this TGF beta and the glycans are correlated to each other. So if you need to understand more about their relations, that is how this TGF beta mediate metastasis through this glycan interaction, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.